Thanks, Amit, for the introduction, and thanks for the invitation. It's a real pleasure for me to be here, and this talk, to be completely honest and open, it catches me in transition time where I'm kind of, I've talked about the results I obtained in my postdoc for too many times, but on the other hand, the results of the new lab are not necessarily mature enough to build a story. So this will be kind of a talk with a few parts. In the first part, I will focus on the results of my postdoc. Maybe some of you have heard that a few years ago when I presented here. But this is a necessary introduction and context for the more recent work. Then in the second part, I will talk about the work I did during the last year of the postdoc, which I didn't have a chance to tell you about, using optogenetics, pharmacogenetics in the context of sleep and anesthesia. And in the final part, I will talk about the new lab's direction with some very preliminary results and maybe some discussion and brainstorming about some future directions, which are very preliminary. Okay, so the overview of the talk. First, I'm going to talk about local sleep oscillations. Then the second part, I will tell you how local sleep-like oscillations may invade the activity of the brain also when we are awake. In the third part, I will talk about the interplay between local states of activity and the global states of behavior and vigilance or brain activity more globally. And finally, I will focus on sensory disconnection during sleep, which is the new focus of the lab. And for some reason, I had a slide with all the students and thank you and funding sources, and Freud would have said something about that, but I can't seem to find it. So at the end, I will pull those thank you slides from a different presentation. All right. So when I began my being interested in sleep, the prevailing story, and it's still the story when you open a textbook about sleep, and this is what I was taught about sleep. Until recently, it was believed that during non-REM sleep, literally all neurons in the cortex and thalamus and other brain regions, such as the basal ganglia and others, alternate between active and inactive states simultaneously. And now I have, I believe, some more context to put this notion into the proper perspective. So this field of examining slow waves, and maybe some of you know it as up and down states, on and off periods of neurons, was put forward by beautiful, technically challenging work of Mircea and colleagues and followers, and other people doing intracellular recordings in vivo. This is one example from Volgoshev, Igor Timofeev and colleagues from a few years ago, in which they amazingly recorded intracellularly from 4 different neurons spread apart by a few millimeters in the cat's brain. And the cat is anesthetized with a mix of ketamine and xylazine. And you can see that during such times, neurons alternate between these inactive periods here, which also correspond to a certain phase of the EEG as recorded outside the brain, and these active periods where there's a heavy synaptic activity, which may or may not be followed by spiking activity. And the rationale of many people in the field was this: if we want to understand sleep, we better focus on non-REM sleep, because it composes 80% of sleep, and also it's the most well-preserved feature of sleep that's also present in non-mammalian species. Well, non-REM sleep is all about these slow waves. These slow waves also occur very prominently in some types of anesthesia, such as ketamine, xylazine. So if we want to understand sleep, why don't we look at these slow waves in very well-controlled condition of anesthesia? And during these conditions of anesthesia, it seemed that all the neurons are going up and down, going active and inactive at the same time. But we decided to look for possible regional differences and regional complexity in these sleep waves. And before our study, there were a few other hints that some regional complexity may occur in these sleep waves. For example, in one study, from the Wisconsin lab where I did my postdoc. They trained people in a sensory motor task before sleep that's known to heavily rely on these regions in the somatosensory parietal cortex. And then they let the participants sleep spontaneously, and they monitored the amplitude or the strength of their slow waves, of their sleep waves following the task. And they found that the intensity of the sleep waves was more pronounced or maximal roughly over the same locations that presumably were intensely involved in the task. So to make a long story short, they found some evidence that if a certain brain region, a certain population of neurons is very active during wakefulness and possibly involved in plastic processes, then those neurons may show, in a sense, a stronger sleep. And of course, Mother Nature also gave us some examples that not all neurons go up and down necessarily in phase and simultaneously, and maybe the most famous example is the sleep of some marine mammals such as dolphins and seals. And of course, dolphins are not fish. They need to go to the surface of the sea in order to breathe. And through evolution, they developed this mechanism where their two cerebral hemispheres sleep out of phase or in turns. It's like shmira b'tzava. You take 1 to 2 a.m., I take 2 to 3 a.m. And here you can see these Eeg traces from the two cerebral hemispheres as a dolphin sleeps naturally. And these discoveries were made in the late 1970s. So, and by the way, when the right hemisphere sleeps, then the left eye is shut and the left fin is inactive, and the dolphins swim with one fin, just like when I take my kids to Parque Yalcon, they sleep in a spiral, just like we do with a boat, when we row with one. Okay, that's just an anecdote. But at any rate, the point of the matter is that there were some examples that sleep needs not necessarily to be entirely synchronous across the brain. And we decided to study this in more detail by collaborating with Professor Yitzhak Fried, who I imagine you may have heard about his research. He treats epilepsy patients that are pharmacologically resistant, so they don't respond well to the drugs, and they are candidates for potential surgical removal of the epileptogenic brain tissue. And for their clinical diagnosis, the neurosurgeons implant these depth electrodes into various brain regions, typically between 8 and 12 regions simultaneously. And the patients spend about 10 days at a hospital ward while they wait for seizures to occur so that the physicians can pinpoint the origin of their seizures. And this presents a very rare and unique opportunity for us researchers to look at what's happening deep in the brain in humans and to record local field potentials, and in some cases also isolate the spiking activity of individual neurons from the human brain. And in this study, we looked at the sleep of these patients, and we recorded, this is an overview, the two hemispheres from a medial view in which you see the sites of recording. So each purple dot is a location of one electrode. And electrodes were placed in medial temporal lobe structures such as the hippocampus and entorhinal cortex, amygdala, parahippocampal gyrus, but also along the cingulate in the medial prefrontal cortex. And in addition, we also had scalp EEG and monitored eye movements and muscle tone via chin EMG to characterize the stage of sleep as is routinely done in a sleep lab non-invasively. And here you can see what we call a hypnogram. So it's essentially the progression of sleep and wake stages throughout a night of sleep. And I can tell you that we were first very worried generally to study sleep in patients who have severe epilepsy, because epilepsy and sleep may be intimately related. But luckily over time we gained confidence that in many ways the sleep of these patients is within the normal range. So they have mostly deep non-REM sleep early in the night, more and more REM sleep later in the night. This, for example, is a power spectrum of their EEG as a function of different states of sleep. So you can see non-REM sleep is characterized by these slow waves and a lot of action in what we call the sigma band between 10 and 15 hertz. This is the frequency band of sleep spindles, whereas wakefulness and REM sleep are characterized by some theta activity and high frequency activity. So again, to make a long story short, we think it is worthwhile to try and look at sleep of these patients and at least in some cases infer some things about the sleep of the general population, and I will try to convince you that this is worthwhile. So let me show you what brain activity looks like during natural sleep deep in the human brain. So here is a trace of EEG. EEG, just like you would get at a Tel HaShomer sleep lab when a person has sleep apnea, right? And this is 15 seconds, and you can see these slow waves occurring roughly with a period of about 1 second. And this is what the depth EEG, or the intracranial EEG, looks from one particular brain region, in this case the right entorhinal cortex. You can see that it also shows these slow waves that have, by and large, an opposite polarity. So when the EEG on the scalp shows this negative peak, the EEG deep in the brain would show a positive peak. And when we look at the activity recorded from the microwires, here you can see some multi-unit activity traces, and here we can see the raster or binary spike train of 6 well-isolated units in the right entorhinal cortex. So what can we see? We see that basically, as was found in animals and as is described in all the textbooks, these slow waves of sleep reflect an alteration between active periods, let's call them on states, and you can see that during these times the neurons spike with firing rates that are perfectly comparable to those occurring during wakefulness. And alternatively, there are these periods of silence where literally no spiking activity occurs, and this goes hand in hand with a positive peak in the depth of the brain and with a negative peak on the scalp. And when we look, we developed an algorithm to automatically detect these waves, and when you look on average, for example, an average here of 600 neurons, you can see that these positivities in the depth of the brain go hand in hand with a reduction of firing, and conversely, these negativities in the depth of the brain go hand in hand with increased firing rate. So far, it's the same story as we've seen in animal studies. So neurons go on and off when the brain is in deep sleep. And here is one example of, so far we've seen one brain region. Now when we look at 5 different brain regions, in this particular example, which represents the deepest sleep occurring very early in the night, what was usually historically referred to as stage 4 and 4 sleep, the deepest phase of non-REM sleep, you can see that in this sleep, different parts of the brain alternate between activity and inactivity relatively in phase. So for example, if we take this particular example, you can see that when there's a negative peak on the scalp, most brain regions show this period of silence exemplified by this white vertical bar. But the truth of the matter is that this image, which is what you find in all textbooks about sleep, was not what we typically found. This was the exception rather than the rule. It actually took us 2 weeks to come up with this one example in one patient. Instead, what we saw all the time when just browsing through our raw data was something different that we, with time, coined local slow waves. And this is just one example that helps illustrate the phenomena. Here you can see recordings from the left posterior cingulate cortex and the right posterior cingulate cortex and the scalp EEG. The first thing to notice, which is important, is that during this time the scalp EEG only shows modest deflections. It doesn't show these massive slow waves. And if any of you have ever looked at EEG of natural sleep, not anesthesia, for example in a sleep lab, you would know that these massive slow waves actually they come and go. Most of non-REM sleep, and typically later in the night, the EEG has many periods where it's relatively flat and doesn't show these dramatic slow waves all the time. And what happens deep in the brain during these times, it turns out that one region can go into this island of inactivity, let's call it an off period, while a different brain region is perfectly active. And a few seconds later, this region actually goes into an off period while this region remains perfectly active. So in simple words, we found that slow waves of sleep often happen out of phase between different brain structure, right? So that it's not like the entire brain is going like this. It could be the case that one region is off, the other one is on, then that one is on, and that one is off. And we wanted to quantify, are most slow waves synchronous or in phase, or are most waves local or out of phase? And when we quantify this, you can see this distribution reflects what we call involvement. What is the percent of brain regions showing one individual wave at the same time? And you can see that this distribution is really skewed to the left. In other words, your average wave of sleep entails about 25 to 30%, can be observed in about 25-30% of the brain. So most waves of sleep, they happen in a chunk of the brain, but not necessarily across the entire brain. These global waves that correspond to waves that are found in 80, 90, 100% of electrodes are actually very rare and were hard to find in the data. Interestingly, another very important sleep oscillation, which is called sleep spindles, we also found that sleep spindles could occur out of phase between different brain structures. And this was even more surprising. And the reason is that we know quite a bit about the mechanism of generation of sleep spindles, and it involves this tango dance between the thalamus, the reticular thalamic nucleus, and the cortex. And in contrast to slow waves, which can happen also in an isolated cortical slab, now the fact that sleep spindles could occur locally means that some part of the thalamus can generate a spindle with some part of the cortex, while a different part of the thalamus is doing something different with a different part of the cortex. So it has a different meaning. Here you can see the sleep spindle oscillation of about 10 to 15 hertz in the right orbitofrontal cortex, and this is in spectrogram form. And you can see that spindles can happen in one region with only a weak trace in the other region, then a spindle here, you can barely see it here, then a spindle here, and you can barely see it there. So we were surprised that not only slow waves, but also sleep spindles may occur in such a local fashion. Importantly, this was a strange feeling as a postdoc. I was both very excited by these results, but also very nervous about these results, because these patients have severe epilepsy that can be focal. Now, if these waves are pathological rather than physiological, then it becomes trivial that one part of the brain shows some epilepsy-like slow wave activity, while a different brain region is doing something different. So we couldn't decide to what extent it's fair to generalize these results to the healthy population. And only when we replicated this in rodents that don't have epilepsy were we confident enough to go out and tell that story. So this is an example here in a rat. You can see the EEG on the top, local field potentials from two brain regions, in this case the auditory cortex and the frontal lobe. Here you can see the multi-unit activity in the auditory cortex and in the frontal lobe, and these raster plots showing putative action potential. So this is a screenshot from our electrophysiology software. There's no analysis. There's no spike sorting. It's the raw data, and I like it because I think that it shows that you don't even need fancy statistics or anything. You can just literally see. Can you see this from the back? You can see these vertical black stripes, which show periods of silence, and they occur generally at different times in the two brain regions. Sometimes they occur simultaneously, but most of the time they just occur whenever. So it's not necessarily true that neurons in different parts of the brain go active and inactive at the same time. So that was a short introductory, and I tried to skim quickly through this because some of you have already heard me talk about this, and I want to move on to more novel findings. The second part of the talk, I will describe to you that these local islands of silence, not only do they occur in sleep, they can also be found in the awake brain when animals and people become very tired and sleep deprived. And this is work that was led by Vlad Vyazovsky, with whom I worked at my postdoc. And what we did is we deprived rats for 4 hours past their normal bedtime. So they are tired, but it's not the end of the world. It's like going to sleep at 3 a.m. or something, right? How do we do this, by the way? We don't touch them. That's stressful. What we do is we take a toy from McDonald's kids meal, and we just put it in the cage. Animals, they like their habits. Why do we do that, by the way? Because, McDonald's are afraid that we'll sue them for a screw that may come off and a toddler may swallow it. So they are actually the safest objects to let animals fiddle with. So you just take one of those pink elephants from, Ice Age 3, you put it in the cage. And animals, it's a big change in their environment. They explore, they sniff the object, they're very excited. And every 20 minutes or so, you need to replace it by a different object. And we do this for 4 hours. And while we do this, we recorded the extracellular activity simultaneously in the frontal lobe and in the parietal lobe. And this is just a snapshot to show you the typical pattern of activity in a small population of about 7-8 neurons. So before sleep deprivation, this is what it would look like. And after sleep deprivation, we started to see these short intervals of about 100 milliseconds. So they were shorter than what we found during sleep, but they sort of had the look and feel of off periods or inactive periods that we were used to seeing during sleep. And what was interesting is, just like the case was during sleep, also when the animals were awake, some of these events were global. Some of this inactivity was across different lobes, but most of these off periods were occurring in one region and not the other. And importantly, as we kept animals awake for longer and longer periods, we found more and more of these off periods. Of course, neurons can be silent for 100 milliseconds regardless of sleep and sleepiness, right? You can play a sound that doesn't fall in the receptive field of an A1 neuron, and it may not fire for 100 milliseconds. That's no news, right? But if these intervals of silence grow more and more probable as the animal is kept awake for longer and longer, then this is an indication that it has to do with sleepiness. And yes? So that's a good question. In this study, we only recorded from these 2 locations. In other studies, we've seen this also in the auditory cortex in other studies. So far, in every region that we've looked at in the cortex and in some thalamic nuclei, we have seen this observation, but it is, I don't know about it. It's a very good question. It's a very good question. What we also found was that not only do these local off periods occur when we're tired, they have a behavioral significance. They affect our cognitive performance. First of all, we all know this intuitively, right? When we're tired, we don't perform as well. But now we thought that we had a handle on some electrophysiological signature of these moments of sleepiness that may affect our behavior. So we trained our animals in a sugar pellet reaching task. They had to run from one side of the cage to reach for a small sugar pellet about the size of a sucrazit. And it's a very difficult task for rodents. Rodents are not that great with their paws. They're not primates or humans. So sometimes they would do it correctly and sometimes they wouldn't. And we looked at each trial with video and categorized it according to the behavior as either a hit trial or a miss trial. And this is just examples, individual examples, but show in this particular trial, a few hundred milliseconds before the animal missed the sugar pellet, we saw this silence-like mode of activity in the neurons that we thought may be related. So we developed an algorithm to quantify the occurrence of such off periods. And what we found was that in the frontal lobe, in the contralateral motor cortex controlling the paw, before missed trials, we found a significantly higher occurrence of these off periods. But this was not true for hit trials, and it was not true, there was no difference in a parietal region that served as a control. So the way we interpret these results is that when we're tired, and I would like to make one important thing to emphasize. We all know what microsleep is. In the sleep world, we call it microsleep, and in simple Hebrew, we will call it litafetz. We all know what this is, right? And this is a moment when our eyes close, the muscle tone transiently drops, and this can actually be recorded non-invasively with EEG. But I'd like to emphasize that what I'm describing here is something different. The animals are with open eyes. They are performing a task. You cannot see anything different from non-invasive EEG. It's only when you record inside the brain do you notice that different brain regions, the brain is so tired and it needs sleep so badly that some circuits fall offline for brief intervals. And if you happen to be needing your motor cortex at that time, when the motor cortex is offline, then this may obviously impair your performance. This is how we think about these results. And what we were curious is, does something similar happen in the human brain when we're tired? Let's say we rent a car and a nice vacation in Florence, and it's 5 a.m., and we've all been there. The question is, what happens in our brain when we are tired like this? And to better understand the cognitive consequences of sleep deprivation, we went back to the human studies where we could do more delicate cognitive manipulation. So we went back to this population of epilepsy patients, and then the interesting thing for us is that some of these patients are kept awake for 24 hours because they don't have enough seizures. That may seem strange at first, but actually these people have undergone brain surgery and they are just waiting for seizures so the doctors can collect enough data, right? If 5-6 days have passed and they don't have enough seizures, that's bad news. So the neurologists tell them, okay, tonight you're not sleeping. And this is an interesting question on its own. Why does sleep deprivation enhance the probability for seizures, which is sort of known fact for neurologists? But we sort of tapped into that scenario to run the same paradigm in the evening and in the morning after these patients have been awake for 24 hours. So we ran a very simple paradigm. We ran it in 10 patients, comparing the situation before and after sleep deprivation. And what we did is we ran what's called a psychomotor vigilance task. It's a fancy name for the simplest task in the world. You see a picture and you need to respond as quickly as possible. You see a picture, you press. You see a picture, you press. What happens after sleep deprivation? There are these moments that we call lapses in which we present an image and it literally can take the patient 2 or 3 seconds to do anything. And those were the trials that are usually thrown out of analysis, right, in a proper experiment, but we focused and zoomed in on those trials to see what was happening in the brain. And we collected around 1,400 neurons, about half of which were in the medial temporal lobe, and 1 out of 6 of these medial temporal lobe neurons responds well to images of famous people and places. We don't know what exactly it responds to, but for us it was sufficient that it responded robustly. So let me show you some examples of responses. This is a neuron in the anterior hippocampus responding to emotional picture of the 9/11 incidents. Here's another, just to give you a flavor of the responses, here's another neuron in the parahippocampal gyrus responding to some landmarks, here to the Eiffel Tower. Here's yet another neuron responding broadly, with a broad tuning curve, responding with different degrees to different pictures, but massively to this picture of 9/11. Here's a more sparse, selective neuron in the entorhinal cortex that responded also almost exclusively to a picture of a famous actress. And then what we did is we compared the responses of the same neuron to the same picture, but we divided the trials based on behavior. So at some trials, so here you can see a raster plot and a PSTH, and you see these crosses. These crosses show you the exact moment when the patient pressed the button responding to the task, whether this picture was a face or a place. And as you can see, most of the time the patient responded quite quickly, but in some trials, maybe 5 or 6 of the trials, especially in the morning after he's been awake all night, the response was delayed. And when you compare the response of the same neuron to the same picture, when the behavior is fast versus when there were these lapses of attention, you find that during these lapses of attention in this neuron, there's a certain time interval between 200 and 300 milliseconds after stimulus onset that that the neuron is completely silent. You can take the spike response of this neuron during this time and actually predict how quickly, when the patient will press the button on your laptop. And when we looked at different individual neurons, this particular time window between 200 and 300 milliseconds came up time and again as a consistent time window in which we saw differences between good trials and these lapses. So we saw this in different neurons. The problem was that each neuron only had, let's say, 3, 4, 5 trials of these lapses. So it was very difficult to assess the statistical significance. So what we did is what probably most physiologists would call a very crazy and stupid thing to do. We just took all the neurons in the medial temporal lobe. We said, we don't care if you're in the entorhinal cortex, hippocampus, come in. We don't care if you respond to 8 pictures, to 1 picture. We collected all these responses in this massive grand raster plot in PSDH. By the way, when we do this per region, separate by region, you can see a beautiful time delay progression from the anterior fusiform gyrus to the parahippocampal gyrus, entorhinal cortex, and hippocampus. It just follows like a Van Essen anatomy diagram of the visual to hippocampus system. You can see all this in the time delays of the neurons. But more important for our thing, when we did this across all neurons, we finally had enough statistical power to demonstrate a significant difference in firing in this time interval. And what was interesting, at those moments when the neurons responded less strongly, we also saw an increase of power in low frequency activity in the local field potentials. So this band of below 10 hertz or below 8 hertz of slow waves and, let's say, theta had significantly higher amplitude during the lapse trials compared to good trials. So the way we interpret this is, again, as was found in the rats, also in the human brain, some circuits may temporarily go in a sleep-like mode, temporarily go offline when we're tired and sleep-deprived, with slow frequency becoming dominant in the LFP and neurons showing this transient off periods, much like sleep, and this may affect our cognitive performance. Okay, so how are we with time? There's still some time. Good. So now I'd like to tell you about completely new stuff, but I'll take your question first.
I was wondering if you thought about checking or if, for example, things like caffeine will affect the electrical activity in these neurons.
It's a great question. So obviously with epilepsy patients, we can't, for ethical reasons, perform these type of studies, but in the rodents, my colleague Vlad Wiazowski now at Oxford, he's following up on these off periods during wakefulness, and he's trying to better understand the conditions and how they may be affected by pharmacological treatment and stuff like caffeine and a variety of cognitive tasks. So he's at present diving deeper into this world. A big question is whether these off periods are adaptive or maladaptive. In other words, it's true that it's clear that they may affect our behavioral performance, but are they already restoring brain activity as sleep does? That's completely unknown. Now that, I think, is also an interesting question that we still don't understand. Yes?
In the beginning of the talk, you mentioned that in different brain regions, so both the Spindles and also the up and down states are out of phase. So are they actually out of phase in the sense that there's a phase shift and you can align them, or are they independent? Because here it seems like you actually, there was a certain amount of time that took the signal to propagate, but if you line them up, you can see that one happens after the other.
So it's a complex answer. It's a good question, and I can't give a very short answer because it's complex. In short, I believe it's mostly independent, but sometimes these slow waves propagate along typical pathways with typical delays, and that very much reflect the connectivity. Some of it is in the paper, some of it I'd be happy to discuss with you at the end. But let's not confuse it with this particular delay, which was the delay of the visually evoked activity, right? So this delay is not the delay of these off periods or sleep waves. Okay, so since I hope I managed to convince you that some of these interesting electrical waves of sleep, like slow waves and spindles, they may happen independently, out of phase in different brain regions, we wanted to take this further, and we wanted to see, so far, different brain regions were in the same state. They were all in a state of non-REM sleep, but their waves were out of phase. But can we push it further to a situation like the dolphin, where one brain region is literally in a different state than a different brain region? Let's say one region is awake, or has a wake-like mode of activity, and a different region is asleep. What would happen? What would happen if the brain is anesthetized and we artificially induced wake-like activity in a certain region of the brain? What would be the functional consequences? So to answer these type of questions, we conducted a series of studies during the last year of my postdoc, and these were, I must acknowledge the contribution of Chad Funk and Alex Rodriguez, and many colleagues also from Japan, and Ofer Izhar from the Weizmann, who really guided us setting up optogenetics. And basically, our motivation was to understand the relation between local activity, global activity, and behavioral states. And our strategy was this. We wanted to use techniques like optogenetics and pharmacogenetics to artificially dissociate the activity of a neuronal population from the global state. And we wanted to do this for two reasons. In the context of anesthesia, we wanted to ask, if we activate a specific population of neurons, can this lead to changes in global activity and behavior? Are there certain brain regions that if we wake them up, this may actually wake the animal up? That was sort of the question we had in mind. And the other question was for sleep, if the brain goes into natural sleep, but we force an island of wake-like activity, will these neurons then become more tired than the rest of the brain that was given the opportunity to sleep naturally? These were the type of questions that we wanted to touch upon. So a few methodological details. We use two techniques. One is called pharmacogenetics, or DREAD. You may have you heard about this? Just so I know this is a good opportunity. So DREAD is a short for designer receptors exclusively activated by designer drugs. It was developed by Brian Roth at Duke, I believe. UNC, I'm sorry, but Carolina, I got that right. So basically they took cholinergic muscarinic receptors, G-coupled protein receptors, and they mutated them and engineered them in the lab to completely lose their affinity to the endogenous acetylcholine. And instead, they now bind to this ligand called CNO, which is inert. It doesn't naturally exist in the brain. So basically, these receptors can sit on neurons. They do absolutely nothing. But if you administer this ligand that's called CNO, after a few minutes it will reach the brain, and when it does so, it will bind to these receptors, and the endogenous intracellular pathways that are coupled to the G protein will take effect. Put simply, this will depolarize the neurons. You are mimicking what would naturally occur when acetylcholine binds to a neuron. The other technique that we used is we used optogenetics, but not the classical channel rhodopsin, which gives you millisecond precise control of activity. We used a novel variant that was developed by Ofer Izhar at his postdoc that's called stable Step-Function Opsin, SSFO. And what's unique about this is with a single pulse of blue light, it opens sodium channels for tens of minutes. So with a single pulse of light, you depolarize neurons for, let's say, 10, 15, 20 minutes. And you can actually shut it off with a pulse of yellow light. And for many questions that have to do with neuronal coding, this is a very bad technique because it has no temporal resolution. But for the questions we were focusing on, such as waking up a brain region for half an hour, this was actually great. You can actually use much weaker light intensity, which is better for the neurons, and it has many advantages. A few other methodological details. We worked with adult male CAMK2 alpha Cree mice. This is maybe the Mizrahi people now can pull out their antennas. And we injected small microliter of adeno-associated virus to induce expression of either these muscarinic-like receptors or these SSFOs and excitatory neurons in the forebrain. And then we waited for a month or two for the virus to express well, and then we conducted our experiments. And we implanted these optrodes, essentially 16-channel microwire arrays, and we glued an optic fiber tip so that this electrode has both an electronics connector and a connector for the optics. And then during our experiment, we collected local field potential, single unit activity, EEG, EMG video, and we observed the behavior. This just shows you expression of these viruses, either for pharmacogenetics or optogenetics. This one fluorescent with red and cherry, and this one you can see the two cerebral hemispheres, so you can see the expression is much stronger in one hemisphere than the other. So the first question we wanted to address with these techniques is this moment when an animal or a person emerge from anesthesia and regain their consciousness. We wanted to experimentally induce this by driving the activity of some select neuronal populations. And of course, this has been done in the past by stimulating different subcortical brain structures, from the classical work of Maruzzi and Magoon in the brainstem, later with optogenetics of either hypocretin or locus coeruleus neurons. And you can even inject nicotine into certain parts of the thalamus, and that would make an anesthetized rat wake up. So this has been done before. But our question was then, can direct activation of cortical neurons also lead to global changes in activity and behavior? And if so, it opens the door to understand if certain neuronal populations, let's say certain layers in the cortex, certain cell types, certain regions may have a more important role in that switch. And that was a part of our motivation. So here's what an experiment looks like. It's very simple. You induce anesthesia either with a gas, in this case isoflurane, or with an injection of ketamine xylazine. You record the activity for about 10-15 minutes of baseline. Then you come in with your manipulation. Either you inject the CNO IP, or you come in with this pulse of blue light, and then you record the effect of your manipulation. Super easy experiment. We did this with 22 sessions and 15 mice. Let me show you what it looks like. Yes?
The CNO, it's a substitute? Is it active, or it's just placebo, or what is it?
So it's some substance that is not naturally inert, so it doesn't exist in the body or the brain as far as we know.
The cell responds as if it is?
The cell responds exactly as if it was hit by a barrage of acetylcholine. So we mimic what would naturally happen in that case in the brain. So here is the activity of anesthetized mouse. In blue on top is the left hemisphere, the control hemisphere, and on the bottom in red is the right hemisphere. This was the hemisphere where we injected the virus. Here you can see typical activity for deep or medium depth isoflurane anesthesia. It's called a burst suppression mode of activity. You see these intervals of suppression, no activity. Then you see a burst that's highly synchronous across the entire brain. Suppression, then you have this burst, suppress, and this just goes on. And after we inject our CNO, we wait 20 minutes. This is what the activity then looks like. The left control hemisphere continues to alternate between suppression and bursts. In a sense, it is active just like during normal anesthesia. But the right hemisphere, where we did our manipulation, look at the neurons here. They are now tonically active in a mode that's typical for wakefulness. Now, if you look very carefully, you may see that the burst is still present here. The burst is superimposed with this tonic mode of activity. We can also achieve a similar phenomena with optogenetics. Here, it happens immediately. You don't have to wait the 6-7 minutes for the CNO to cross the blood-brain barrier and reach the receptors. You press a laser, and then in that same instance, the neurons just go crazy and they fire tonically for tens of minutes. It's a very powerful manipulation, right? The animal is anesthetized. You take a certain passive cortex, and it's like in the Chevrat Chashmal. You turn on the switch, and the neurons go. The interesting thing is, in some cases, now we could start asking questions. For example, does this local island of activity stay local? Does it spread all over the brain? Can it actually affect behavior? And in some cases, we indeed saw that local activity also changed the EEG in distant screws in the frontal lobe. So this was the EEG before pharmacogenetics, and this was the EEG after the pharmacogenetics. So now it's very far away in the brain. So we managed to activate the EEG, which is taken as a sign of cortical arousal. So in this particular example, the local activity spread into this global fire in distant areas of the brain. And we could quantify this. We saw that our manipulation caused the local LSP to change in 90% of channels, but distant EEG, so this is the global activity, to change in 50% of the cases. So we almost always could manipulate local activity. In about half of the cases, this local activity spread and influenced the entire brain. And what was interesting, what is that? Including the other side, including everywhere we could record. We didn't record in 12 different brain regions in the mouse as we did in people, but in very distant EEG screws to the extent that they reflect the activity that is just below the screw. So, and interestingly, in some cases, it also led to behavioral emergence from light anesthesia. So this is like Jesus standing up and starting to walk, and I'll show you some movies which we're sort of enthusiastic about. Hold on. So let me show you, I really like to be able to show raw data when possible. So, hold on. Can you see this? No, this is stuck on, hold on, here I have some technical, whoops. Okay, let me show you what it looks like. See this? Okay. Excuse me for this. Okay, so first, these are all the people involved, and we made a nice film, and this is the film. So let me show you what it. Can you see this from behind? Good. So this is the baseline period. This is the typical activity during isoflurane anesthesia. Here you see the mouse. It's in a gas chamber. That sounds awful, but it's in a chamber where it's under stable equilibrium state of constant anesthesia. So we don't touch it. That's the point. We don't do anything. Here are the Eeg traces. The top two are Eeg. These are local field potentials from the parietal lobe. And these are the multi-unit activity. And here you can see some events that are suspected as action potentials. And this is the EKG. And this just goes on and on for as long as the animal is anesthetized. It's very typical. You see these bursts. And then you see these periods of suppression. Here you see a burst and then a suppression. I'm not sure how well you can see this, but it toggles between burst here and no spikes. A burst and no spikes. A burst, and this just goes on and on. So this is the first movie. Now, the second movie shows the situation. Does it show the situation? Try again. Okay, this shows 10 minutes after the CNO injection. So this is pharmacogenetics. Same animal, same brain region, same traces. Look how different it is now. The EEG is flattened. This is what an EEG of an awake animal would look like. And the local field potentials are completely flat, driven by high frequency activity, and the neurons are now firing tonically. Look at this red neuron here. It doesn't have these long intervals of silence that we were seeing when the animal was anesthetized. So now is a situation where we locally and to some extent globally woke up the brain. But still behaviorally, the animal is immobile, right? It's almost, if you wish, an experimental model for a locked-in patient, if you will, right? With awake-like brain activity in an immobilized body, or maybe something similar to what may happen in REM sleep. That's a different discussion. But here's what we were particularly excited about. This happened spontaneously in some cases, in this case 40 minutes after we did our manipulation. So again, this region is active. You can see the spikes, tonic spiking activity. And then we started to observe spontaneous behavior. At first, it's like these little twitches of the mouse, which may or may not reflect purposeful behavior. Well, we can twitch also when we're anesthetized. That's not necessarily a huge deal. But here, right here, we see what we call in the anesthesia field a righting reflex. So the animal spontaneously rights itself and stands on its 4 feet. And that is usually in animal research of anesthesia, with all the caveats, taken as a sign of purposeful behavior. And as far as we can create analogies between animals and people in terms of regaining consciousness, this is the closest that we see that an animal may have gained back its consciousness, because at least it's displaying some purposeful behavior. And we did this by forcing tonic activity in a select population of the cortex. So now that we see that, so to be honest, this is just like a proof of concept. It opens more questions than it gives answers. But it's a capability that we can now use to ask questions that we couldn't ask before. Now, let's say in this brain region and in these neurons, let's say 20% of the time it will make the mouse wake up and walk. If we would do it in a different brain region, in a different layer of the cortex, in different types of cells, maybe we could get a different probability. Maybe we can find a population of neurons that would wake up the animal 90% of the time. Maybe because those neurons are connected differently. Maybe because they secrete some other neurotransmitter, neuromodulator. So it opens a lot of questions to do something in a more fine-grained manner than was possible before. How are we with time? Not much time. All right. So I think I need to wrap up within a slide or two, or not? Hamid? Be honest. No problem. Yeah, so I will say that I'll just give you it's fine. I always put too much stuff in my presentation, so this is let me just try to overcome some technicalities here. So I will just tell you the new focus, the focus of our new lab is the issue. I'll just give you a teaser. Is the issue of sensory disconnection from the external environment in sleep and anesthesia. Sorry. It worked? So let me actually, no, let me talk for the last 2 minutes about something different. Okay, thank you. So now we did the same exact experiment, but instead of anesthesia, we did it with natural sleep. Imagine you have this mouse, it naturally goes to sleep. You have these slow waves. Now we induce tonic activity in an island of neurons. We do this, let's say, for an hour. Then we let the mouse be. And we follow its subsequent sleep, its recovery sleep. And the question is, these neurons that we kept in a wake-like mode artificially, did they become more tired than other neurons? What is actually the basic unit or basic spatial scale of being tired. Does a person get tired? Does a brain get tired? Does a column get tired? Does one neuron get tired? This is the type of question that we try to touch upon with this tool. And so let me just show you some results. Here is the sort of same experiment like I showed you with anesthesia, but here you have one hemisphere in natural sleep. See neurons going up and down, up and down, up and down with these slow waves. The other hemisphere neurons are just tonically active. Then we let the animal be. And in this one, this is an example of one neuron that we artificially kept awake. So this is a control neuron. And here on the y-axis, you see the percent of time that it was in these off periods. This is natural sleep. Sleep starts with a neuron being about 20% off, and then it progresses, it dissipates into about 5%. This neuron, we didn't let it sleep. We didn't let it go into inactive period. So this is, while we were working with a laser, by definition, it's 0% inactive. And then when we let it be, it showed this rebound. So this particular neuron that was kept active later showed more prolonged off periods, and in a sense, a stronger signature of sleep than the other neurons. And now we can do experiments where we manipulate the light source of the laser beam and the configuration of the electrodes so that we can actually measure in millimeters or microns the scale of becoming tired. And we think it's something exciting to look at. Okay, so on the interplay between sensory systems such as the auditory system and states such as sleep and wakefulness, this would have to wait for maybe the next talk in 3 years' time, when I hope to have more mature results from the new lab. So we're curious about what makes us disconnected from the external world during sleep. For a long time, the story was that the thalamus acts like a gate. It doesn't send stuff to the cortex during sleep and anesthesia. I personally believe that's an insufficient explanation. For example, when you record an A1 of a rat in natural sleep, the responses are perfectly the same. And we do this in different ways. For example, now also in the operating room with these epilepsy patients, at the end of the 10 days, we pull out the electrodes. They are actually anesthetized while we play sounds to them with iPhone earphones, right? So they go into brain surgery with earphones, and we play sounds as they gradually lose consciousness during surgery. And in the one time, this is work by Aaron Krom and Amit Malmstein, in the one time that we were able to record from A1 of such a patient, individual neurons, they seem to respond perfectly, in a perfectly comparable manner. So we believe the action or the interesting differences between sleep and wake happened beyond the primary sensory regions. If you think about it, it makes sense. We've been recording in anesthetized animals for tens of years. We've been seeing beautiful sensory responses, right? So there's something to think about and maybe to discuss for those who are interested. We personally believe that the action may have to do with the locus coeruleus-noradrenaline system, so we're doing a bunch of studies to look at that. And I'll leave you with a final riddle. Do the eye movements of rapid eye movement sleep, do they have anything to do with our mental dream? When we move our eyes, is it like we're seeing a new image? That's a basic question, right? We have a state where we experience images and we move our eyes. It's so intuitive. You would think maybe every time we move our eyes, we see a new image. Is it so? Is it not so? We have some new data to suggest that whenever the eyes move, those neurons that respond to pictures have a burst of activity that looks just like when you actually look at a picture. So we think that's exciting, but some things don't work out. For example, congenitally blind people may move their eyes too. That doesn't mean they're experiencing visual dreams. It's a puzzle. I'd love to discuss that with you. Okay, so thanks for your attention. Thank you very much.
Okay, questions? So going back to your sleep, your initial microsleep evidence, I mean, is there any evidence for these microsleep episodes to be actually causally, I mean, do they have any causal effect on behavior? I mean, if I took the same, let's say, circuit before and after it went through this microsleep episode, will it be more effective?
We have no idea. It's an open question. In other words, are these local off periods, do they have some adaptive value along the lines that sleep has? We simply don't know. We simply don't know. And once again, I have sort of shifted focus from that finding, but a good friend and colleague of mine in Oxford, I'll be happy to make the connection. He's really diving into that specific question right now. I don't think anyone knows yet. Yes?
There's a phenomenon of vocal hallucination the second before a person goes to sleep, even in healthy people. Was it reported in the patients you studied or measured?
It's a great question. So when we fall asleep, we go through these hypnagogic hallucinations. Maybe some of you, you played Candy Crush all day. You close your eyes. You see these colorful circles. Unfortunately, we were unable to collect proper reports from these patients. I wish also when we were lucky enough to record during REM sleep and even the eye movements, we didn't really have a chance to ask them about their dreams. You must understand the situation. They're stressed in a hospital with their mom, waiting for the brain surgery coming in 2 days. We come in with a huge rack of Neuralinks or whatnot, and when we paste additional electrodes to record their eye movements and the chin, and then you feel quite uncomfortable, sort of going, what did you just dream about? So it's fascinating, but quite challenging to actually do. That would be a fascinating thing to look at.
Do animals wake or leave anesthesia earlier after inducing the tonic behavior?
They may. So the question was, when we induce this island of tonic activity, do animals wake up from natural sleep earlier? From anesthesia, I've showed you some examples that it may occur. In natural sleep, I'm not sure we have quantified properly, because rodents have quite fragmented sleep. So after a few minutes, they'll wake up even regardless of an experiment. It's not like humans where they sleep for 10 hours. So you really need to quantify, do they wake up, let's say, in 40 seconds? Is that above and beyond them waking up spontaneously after 40 seconds? We haven't looked at it, but I can tell you the story is a bit more complex than obviously can be described in this type of lecture. Sometimes you overdo the manipulation. You can cause seizure-like activity. You can cause spreading depression. You can cause animals waking up. And you really need to titrate your manipulation to a physiological-like range in order to draw proper conclusions. So it's a good question, and one that brings about a huge box of confounding factors that I didn't discuss in this short presentation.
So I follow up on this.
Yes?
Is there a animal model for locked-in syndrome?
Well, this was just a sort of as a teaser to make people engaged. I'm glad. Well, I would state it in a more precise manner. We can certainly, at will now, in the lab, make an animal show wake-like electrical activity with prolonged immobility. What does that mean is a complex issue. But the interesting thing is that this wake-like electrical activity is happening in a biochemical bath that is typical for sleep. This is a thing that I didn't discuss, right? Usually, we either have tonic activity with serotonin, acetylcholine, noradrenaline, histamine, hypocretin, and all that, or we have low levels of these modulators, and the neurons go up and down. Here we have a new situation where we can force the neurons to have tonic activity, but the subcortical systems are still operating in a low mode, typical for sleep and anesthesia. I'm not sure that is a good model for a locked-in patient, but it's an interesting possibility. It opens some new questions for us as researchers.
Okay, Tamara,
We started the lecture with the example of the dolphin, and I think, I'm not sure, but I saw that even this hemisphere that is awake, not really awake, it's just doing the reflex thing of going up, breathe, and down. So it's not like full wakefulness. It's a functional role that the dolphin needs to do during the sleeping. I'm not sure. But it leads me to ask about, you're talking about the population that are off, the population that are on. Do you think that the ones that are on are really full on, or they have like a different, also they're not really awake?
So it's a good, once again, those are deep, good questions that I don't have decisive answers for. But all I can say is that to move a fin, I don't think you need an entire cerebral hemisphere to be tonically active. That doesn't, intuitively for me, that doesn't make sense. You can have, and you have experts here in brainstem locomotion, generators and whatnot. An animal can swim to grasp for air. I don't think it necessarily would need an entire cerebral hemisphere being tonically active. So in my mind, I would say it's wake-like, and it doesn't serve only the purpose of swimming for air. That would be my guess.
And well, returning to the experiments of waking up the cortex, your Jesus experiment, which makes people think maybe you could do something like that to wake up comatose patients. There's one caveat that I've always been worried about. That same mouse, if you pinched his tail, he would also become lighter and might actually get up on his feet. Do you have any way of distinguishing the possibility that your cortical stimulation is arousing the cortex versus your cortical stimulation is causing pain, which is very similar to pinching his tail?
At present, I don't think we can dissociate those two possible interpretations. My intuition is that both pain and this weird artificial cortical manipulation both have a common end target of exciting the arousal centers of the brain, be it the locus coeruleus or the reticular formation. That wakes actually up the animal. I don't think that the neurons in the cortex firing makes the animal stand up. If the cortical stimulation is strong enough, via projections, it would stimulate the arousal centers, possibly, but in a manner akin to how a painful stimulus does it.
So you don't think your dread is actually turning on the cortical neurons directly, but rather it's turning them on, which is now sending a signal subcortically, which is coming back to turn on the cortex.
So no, let me make it more clear. So I do think we are directly waking up the cortical neurons. I don't think that is the activity that makes the animal stand up. I think that between the cortical neurons being tonically active and the animal standing up is a complex chain of events that I would guess involves also subcortical structures and arousal centers. So, but we don't really know. And it would be, but these experiments can be done. We could induce tonic activity in the cortex while inhibiting, let's say with optogenetics or DREADS, different subcortical arousal centers and put that to the test, but we haven't done so yet.